Hi everyone, this is Staar number 3, cells part 2. The first section is for photosynthesis and cellular respiration. Remember, photo means light, synthesis means to build or to create. So I'm using light, or plants are using light to build glucose. This process happens in the organelle, the chloroplast. Remember, who has chloroplast? That would be plants. So if we look at this picture right here, this is a boxy plant cell, and the lit up green organelles are going to be chloroplast. There are two parts to photosynthesis. The first part is called light dependent, so I depend on light. For light dependent, what's going to happen is in this organelle of the chloroplast, we've got tiny structures called thylakoids. And these little pancake-looking guys, what they do is they absorb solar energy, they absorb light, they take in water. And what they do is they basically use the water to pull hydrogens off of it. The hydrogens and the energy from the light gets stored in an electron carrier called NADPH. And then we also make a little bit of ATP. Why do I need those two processes? I need those two processes to power the second part of photosynthesis, which we'll talk about in a second. Alrighty, so now that I've used light, I've used water, I've pulled off the hydrogens and made my electron carrier, what am I going to do with the leftover oxygen? Plants don't need that oxygen, they basically just breathe it out through the openings in the leaves. So that oxygen will leave, they don't use it, that's one of the products. Alrighty, the second part of photosynthesis is light independent. I don't need light. Where does this occur? This occurs in the empty space of the chloroplast called the stroma. So the empty space that's not a thylakoid, called the stroma, is where this process happens. What do I do in this process? I take carbon dioxide from the air, I use the electron carrier and the ATP I made earlier in light dependent, and I basically use that energy from these two guys to push 6 carbon molecules and 12 hydrogens and 6 oxygens together to make glucose. So I'm taking the carbon from carbon dioxide, I'm using the energy from the first step and pushing it together to store it in the form of glucose. The equation that you need to know for this process is down here at the bottom. So I take in carbon dioxide, water plus solar energy. I get out oxygen and glucose, C6H12O6. You do need to remember that equation. Alrighty, the second picture in the same section, we're in the same section right now, is for cellular respiration. Cellular respiration happens in the organelle called the mitochondria. Who has mitochondria? Plants and animals both have this. So in other words, if I ask you who carries out cellular respiration, that would be both plants and animals. There are 3 steps to this process. The first step is going to be called glycolysis. Glycolysis happens in the cytoplasm of the plant. Alrighty, so what happens in glycolysis? So lysis means to break, glyco refers to glucose. So what basically happens in glycolysis, we take our glucose molecule, we take our sugar, and we're going to lyse it, we're going to break it, and we literally just break it in half. And when I break it in half, that means I get 2 molecules and each side has 3 sugars in it. So step number 1, breaking the glucose. This happens outside of the mitochondria in the cytoplasm. Step 2 is called the Krebs cycle. Krebs cycle happens in the middle of the mitochondria. And what I'm doing is I'm taking that broken down glucose and I'm going to actually break it down even more. So when I break it down even more, I'm breathing out CO2. Where did that CO2 come from? From fully breaking down glucose. How did I power this process? Krebs cycle is powered because in glycolysis, I made NADH. Remember, NADH is an electron carrier. So basically in glycolysis, I made an electron carrier. I used that electron carrier in the Krebs cycle. So now that I've broken down glucose completely and it's back to carbon dioxide, I've made 2 ATP in glycolysis, 2 ATP in Krebs, and what the most important thing I get out of Krebs that's happening in the matrix is I actually get more electron carriers. So after Krebs, these electron carriers are going to be used in our third part, the electron transport chain, also known as ETC. Where does ETC happen? It happens in between the inner and outer membrane. So this that I'm drawing right now, this is going to be the inner membrane, and then the solid outer line is my outer membrane. So in this space right here where I'm putting dots, that's where this process is actually happening, okay? What are we doing in this process? We're taking those electron carriers and we're removing the hydrogens. When we move those hydrogens across the membrane, it produces ATP. I make 34 ATP out of electron transport chain. But I don't want all these hydrogens to build up on the inner membrane space. So in order to get rid of them, I'm breathing in oxygen gas. I'm using that oxygen gas, combining it with hydrogens, and I get water out of this. So the equation for cellular respiration is I breathe in oxygen, I take in glucose, and what do I get? Carbon dioxide, water, and ATP. This reaction is almost identically backwards to the process of photosynthesis. So remember, for photosynthesis, I have to give plants carbon dioxide, water, and solar energy. Here, I'm getting out carbon dioxide, water, and ATP. ATP is just a form of energy. Is it identical to solar? No, but it is the opposite effect of it. Remember, mitochondria, they are located in both plants and animals. So who does cellular respiration? Both plants and animals. And the last thing is aerobic versus anaerobic. Aerobic means it requires oxygen. Anaerobic does not require oxygen. So if I ask what type of organism are we, we're going to be aerobic. That's why, for example, when you can't breathe, that's why you actually die or your cells start to die for the simple reason that we require oxygen gas to power the electron transport chain. So if you don't get oxygen, if you're suffocating, then you won't be able to produce ATP through the process of cellular respiration. Well, if I can't produce ATP, I can't power my muscles, I can't power my cells, and so they will eventually start to die. Alrighty, the next section is for the cell cycle. The cell cycle has 4 major parts, G1S, G2, M, C. I'm sorry, that's 5 major parts. Those are all the parts that a cell must go through to complete the cell cycle and produce 2 identical diploid somatic cells. And remember, the word somatic means body. So basically, I'm producing 2 identical body cells. And again, the steps that I use to go through them are right here. A little way to remember this is go Santa go, Merry Christmas. So as long as you remember that, remember we are still like 20-something days away from Staar. As long as you remember go Santa go, Merry Christmas, you'll be able to remember the order of the cell cycle. Why are we going through the cell cycle? The cell cycle is for growth and repair. In other words, I'm doing this process so that if I need to grow larger in size, I need to grow taller, for example, I want to copy my cells, make identical ones so that I can grow larger. To repair, if I lose skin cells, hair, whatever it might be, I need to repair that with new skin cells, with new cells, and I do that by going through the cell cycle. Alrighty, so G1, S, and G2 can all be condensed by calling it interphase. So interphase is going to be these 3 parts of the cell cycle right here. For the S phase, there's something we do need to talk about how it happens. For S phase, what I'm doing is I'm copying my DNA. So G1, I'm just copying organelles. S, I'm actually copying my DNA. Why do I do this? I do this so that my daughter cells can have the same copy of DNA. So I have to copy it, just like I copy papers on a copy machine. How does this happen? For the S phase, for DNA replication, we've got 2 major enzymes working. There are a bunch more, but we're going to focus only on 2. The first one is helicase. What does helicase do? Helicase unwinds DNA. In other words, it breaks hydrogen bonds. The second enzyme we're going to focus on is DNA polymerase. DNA polymerase's job is now that the DNA has been unwound, I can actually read both sides of DNA and make 2 identical double helix. So if you notice in this picture right here, I've got 1 double helix forming, 2nd double helix forming, and it came from this one original. So this double helix right here will go into one daughter cell, this one will go into the other daughter cell. So they both have identical copies of DNA. Alrighty, going back to interphase, so we have copied organelles, we have copied DNA, and then we go back to G2. G2 is basically just any checks, make any fixes to any errors that happened. Let's look at the pictures though. So notice in G1, I've got a mom and dad copy of this chromosome. After S, I still have a mom and dad copy, but notice it's in the little X shape. So just as a reminder, this is one chromosome from one chromosome from dad, and this one is from mom. Well, after I go through S, what happens is I take the copy of the dad one, and I combine it with the second one. So this chromosome and this one are still identical. Each half of this chromosome is the same. What do we call each half? We call them chromatids. Okay, so chromatids. So this is one chromatid, that's my second chromatid. And I do that same thing for the dad chromosome. Okay, so for the mom and dad chromosome, they get copied and they've got identical attachments to them. What do I call these two together? So these two right here are called homologous chromosomes. That's an O right there. Homologous chromosomes. These two are also called homologous chromosomes. So it doesn't matter if it's in the little X shape or if it's single line. As long as I'm talking about the mom and dad copy of the same type of chromosome, then it's going to be homologous. Mom and dad copy of the same type of chromosome, homologous. So let's look back at the picture. Right here, it's a mom and dad copy of the same chromosome, so that is a homologous pair. Right here in the yellow X's, that's a homologous pair because it's a mom and dad copy of the same chromosome. Alrighty, so let's go back to our picture. The next stage after interphase is going to be mitosis itself. So right here, mitosis. Remember the stages of mitosis are pro, meta, ana, telo. So the easiest way to remember that is please marry Anna today. And then at the very end of this process, we have cytokinesis. Cytokinesis is where my cell actually separates itself into two separate cells. Cancer is going to be when a cell goes through the cell cycle repeatedly uncontrolled or unregulated. So if I go through the cell cycle way more than I need to, I'm going to actually produce a clump of cells that are unnecessary, called a tumor. Tumors are harmful because basically what they do is they take away all the nutrients and everything that your healthy, normal cells needed. And so it's very harmful to your body. It's basically like inviting a little parasite, but it's actually your own cells. What causes cancer? Mutations in your DNA, chemicals, things like that will cause cancer. Really quickly, this is what mitosis looks like when they actually show you real pictures. So up here, this first picture that we're looking at, notice my nucleus is still together and all the DNA is just one solid little blob. So this is going to be interphase, so this first picture. The second picture, my nucleus is breaking down and all the little red lines you see in the middle, that's my chromosomes. So this is going to be prophase. The next one, notice all of them are lined up along the middle. This is the middle of the cell, and all of those chromosomes are lined up in the middle. So meta, middle. The next one, anaphase. My chromosomes have been pulled apart into two halves. So ana, apart. And the last one, telophase. I have two nuclei formed, telophase, two nuclei. If we look at the picture that you have on your notes, this right here is not the mitosis yet. This is actually still interphase. This in particular is S phase. This is the first part of mitosis. They skip prophase in this picture. They just show meta, ana, and then this last one right here would be cytokinesis / telophase. Remember, I do get two diploid cells. If you don't remember what the word diploid means two copies of each chromosome. In other words, homologous pairs. So notice right here, I've got two chromosomes that are the same length. Two chromosomes that are the same length, those are diploid. Alrighty, the last section of our notes is going to be meiosis. For meiosis, it actually has two parts of cell division. What you need to know about meiosis, it is the process to produce 4 gametes. These gametes are all going to be unique, and these gametes are all going to be haploid. Again, the word haploid means that they only have one copy of each chromosome. So let's practice this really quickly. If I were to show you a cell that looked like this, draw that one more time. If I were to show you a cell that looked like this, it has 2 tall chromosomes and 2 small chromosomes. Well, the 2 tall ones are homologous, the 2 small ones are homologous. So this would be diploid, 2N, diploid. If I showed you a cell that only had 1 tall one and 1 small one, that is not a homologous pair, so that would be haploid. The same thing if I showed you this cell. Oops, lost my chromosome. Alrighty, so I've got 2 tall ones, 2 small ones. Again, that is diploid. And this one right here would be haploid. Okay, so keep that in mind. Make sure you know the difference between haploid and diploid. Haploid means it only has one copy of each chromosome. Diploid means it has two copies of each chromosome. Alrighty, so for meiosis 1, what we're looking at basically, the most important thing is that for meiosis, it follows the same step. It goes from interphase to meiosis 1, cytokinesis, and then it will go through a weird meiosis 2, and cytokinesis. But overall, the process is the same. So we go through prophase, anaphase, metaphase. I'm sorry, I did that backwards. That's okay. Metaphase, anaphase, and then we go through telophase or cytokinesis. But the most important thing you should pay attention to is in prophase 1 right here, we do something called crossing over. What is crossing over? Between my homologous chromosomes, so between my mom and dad copy of the chromosomes, I'm swapping genes. Why do I want to do this? Because it gives me unique cells at the end. Remember, all these gametes are going to be unique. So let's go back. I've got prophase, my homologous chromosomes line up in the middle, metaphase, my homologous chromosomes get pulled apart, anaphase. And then we start meiosis 2. In meiosis 2, again, I go through prophase, and then I'm going to go through metaphase, where my chromosomes line up in the middle, anaphase, my sister chromatids get pulled apart, and then telophase / cytokinesis, where my cell actually splits into 2. Again, these are 4 haploid cells. An easy way to remember this is what I always use. My toe is growing. So for mitosis, it is for growth. And if I want my toe to grow larger, I want all of it to look the same. I don't want it to look different parts. So for mitosis, my toe is growing. That'll help you remember that.